Hello everyone, welcome back to the class. In our previous class, we discussed about the role of S-Cdk. The S-Cdk is commonly referred to as mitosis promoting factor or maturation promoting factor, that is MPF. It is synthesized during S phase and G2 phase. S-Cdk complex has many functions like they induce chromosome condensation, breakdown of nuclear envelope, assembly of mitotic spindle apparatus, alignment of condensed chromosomes at metaphase plates. So all these functions of S-Cdk complex is dependent on the phosphorylation activity of S-Cdk. So it phosphorylates wide range of proteins. It phosphorylates lamin proteins in order to stabilizing the network under nuclear membrane, which helps in breakdown of nuclear envelope. It phosphorylates condensin proteins which induce chromosome condensation. Condensin is a type of protein which is highly conserved, which is a 5 subunit protein complex. Among 5, 2 SMC proteins are important, SMC2 and SMC4. Both these has a fundamental role in structural and functional organization of chromosomes. It is the key driver of mitotic chromosome condensation. So condensin is phosphorylated step by step by Cyclin-Dependent Polo Aurora B kinase in a manner that correlates with changes in its localization, dynamics and supercoiling activity. And also we discussed about how S-Cdk influences the assembly of mitotic spindle. It is done by phosphorylating proteins that regulate microtubule behavior. These proteins are microtubule dependent motor proteins and microtubule associated proteins, which is in short we call it as MAPs. So S-Cdk phosphorylates MAPs for accurate separation of chromosomes during mitosis. It helps in proper association of all chromosomes with spindle microtubule. After doing all this, S-Cdk at last activates the APC-BARC to set the stage for progression into anaphase. This S-Cdk not only triggers the pre-initiation stage of metaphase events, or it also help in transition of metaphase to anaphase. APC-BARC here acts on different substrates at different times. So it functions in coordination with two homologous mitotic coactivators, that is CDC20, CDH1. This initiates metaphase-anaphase transition through mediating the ubiquitination and degradation of M-cyclin and securin. As securin holds the enzyme separase. The separase, whenever it is with securin, it is inactive. In order to make it active, so the destruction is much needed. So destruction of securin allows separase to cleave SCC1, which is a subunit of a cohesin which holds sister chromatids together. It prevents premature separation of sister chromatids and exit from the mitosis. APC-BARC is activated by CDC20. The active APC-BARC help in making the inactive separase to active by ubiquitination and degradation process of securin. So this active separase cleaves and dissociates the cohesin in order to pull out the sister chromatids. So the decrease in S-Cdk activity permits the separated chromosome to decondense. Nuclear envelope reforms around the daughter cell nuclei during telophase. Cytoplasm divides at cytokinesis, which yields the two daughter cells. APC-CDH1 is inactive in early mitosis and only becomes active from late mitosis to G1 phase. Destruction of cyclins is very important for exit from mitosis, as it synthesizes synthesis is for entry. So this is the overall overview of the current model for regulation of the eukaryotic cell, starting from G1 to the action of APC-BARC, which is initiating the metaphase and anaphase transition. At all these stages, all the CDKs play an important role. So today we'll be learning about another complex, that is S-Cdk, which controls the DNA duplication in S phase. So it's all begins from the preparation for DNA replication. So the preparation of DNA replication as we are discussing about the S phase, so it is all about DNA replication, where the S phase is predominantly synthesizing the DNA. Okay, so the preparation of DNA replication happens in early G1 itself, at beginning of the G1. It starts by the assembly. It starts by the assembly of PRC. What is this PRC? It is pre-replicative complex. So all the preparation of DNA replication at the beginning in the G1 it begins by the assembly of PRC. Where this assembly takes place? This assembly takes place at the replication origin, or we can say it is a origin site of replication, where the replication begins in the DNA. Okay, so the S-Cdk plays a crucial role. S-Cdk, that is the activation of S-Cdk leads to the formation of many complexes which help in the control synthesis of DNA and replication of DNA. So S-Cdk activation leads to formation of many multi-protein pre-initiation complex. So when what happens when S-Cdk is activated? When S-Cdk is activated, it leads to the formation of multi-protein pre-initiator complex. What is this? What is the function of pre-initiation complex? This unwinds the DNA particularly at origins, and this after unwinding they begin the replication. So this is the function of pre-initiation complex. So it all started by the activation of S-Cdk. S-Cdk activation led to formation of multi-protein pre-initiation complex, and that unwinded the DNA at the origins, and it began the process of DNA replication. The key player involved in this process is a large multi-protein complex that is known as ORC. Now what is this ORC? ORC is nothing but origin recognition complex. So in order to bind the pre-replicative complex at the replication origin, it is crucial to first find out the origin of replication. So this ORC plays a important role in recognition of origin. Okay, it binds to the origins of replication. Where does it bind? It binds to ORI region or origin of replication. So how does it do? So in early G1 phase, there are co-activators, that is CDC6 and CDT1. So these are the co-activators which is associated with this ORC. So in order to activate the ORC, which is the origin recognition complex, the co-activators CDC6 and CDT1 should associate with it. So now the ORC is active. So this CDC6 binding with the ORC helps the ORC to bind or assemble with another protein, that is MCM protein. MCM full form is mini chromosome maintenance protein. Mini chromosome maintenance protein. So the full form of MCM is mini chromosome maintenance protein, which acts as a helicase. This acts as a helicase. What is helicase? It unwinds the DNA. It is a MCM is a hexamer of 6 related polypeptides. So it is a hexamer made up of 6 related polypeptides, that is MCM2 to 7. MCM2 to 7, that forms a ring structure. So you can see the MCM along with ORC and ORC associated with CDC6, CDT1, CDT. This is CDT1. This assembly is called as pre-replicative complex. Pre-replicative complex. So what are the components present in the pre-replicative complex? That is CDC6, CDT1 associated with ORC, and ORC resulting the assembly with MCM protein. All these together is known as pre-replicative complex. So the formation of pre-replicative complex will give the license to the origin. So the origin is now licensed. So origin, consider this region is ORI region of DNA, where the unwind should happen. This pre-replicative complex comes and binds. Remember, what are the components present in the pre-replicative complex? CDC6, CDT1, ORC and MCM. So all these are forming a pre-replicative complex. Okay, so that comes and binds to the origin region, giving a license for the replication. So this MCM protein acts as a licensing factor. So this allow, I'll write it in a different color. So this allow replication to initiate, which is playing a crucial role here, MCM, for initiation of replication. Once the pre-replicative complex has assembled in G1, the replication origin is this replication origin is ready to fire. That means it is ready to replicate. So the activation of S-Cdk, so the activation of S-Cdk is responsible in late, especially the activation of S-Cdk happens in late G1 phase. That helps in, or that is responsible for the triggering, that triggers the assembly of several additional protein complex at the origin, leading to formation of pre-initiation complex. This is not enough. This complex is not enough. There are more proteins complexes which are necessary for activation, which are necessary for assembly, leading to the formation of pre-initiation complex, which will unwind the helix and begins the DNA synthesis at the same time as it initiates DNA replication. So S-Cdk not only activates or not only initiates DNA replication, but also helps in disassembly disassembly of some pre-replicative complex components. Why is it so? So we now know that pre-replicative complex is very much needed for replication. So then why S-Cdk is triggering the disassembly of pre-replicative complex? See, this S-Cdk is helping in disassembly of pre-replicative complex components. Why? Because to reduce or to block we can say, to block re-replication. There are chances for the re-replication of DNA, which will cause a much important error, which will help, or which will destruct the whole cell cycle process. So it blocks the re-replication by how? By destructing the coactivator CDC6. CDC6 is a coactivator which is helping the ORC to bind with the MCM protein, right? So this S-Cdk triggers the disassembly of pre-replicative complex, or it blocks the re-replication by triggering the destruction of CDC. And if there is destruction of CDC6, what about ORC? ORC becomes inactive. So when ORC becomes inactive, will it bind to the, or will it associate with the MCM protein? No. If there is no assembly of MCM protein along with ORC, will that bind with the origin of replication or the region of replication? No. So what will be the result? So it will destruct the entire pre-replicative complex. This is the major thing. So S-Cdk is also triggers the assembly of pre-replicative complex. At the same time, after initiation of DNA replication, after the replication is done, so S-Cdk triggers the disassembly of some pre-replicative complex components as well. See here, control of DNA replication in S phase. The pre-replicative complex assembles in the replication origin, marking the preparation for DNA replication in G1. S-Cdk leads to the formation of the multi-protein pre-initiative complex. So what is this pre-initiative complex? What does it do? It unwinds the DNA at origin and begins the process of DNA replication. The key player are ORC. What is this ORC? Origin of recognition complex or origin recognition complex. It is a large multi-protein complex. It binds to the origin at the early G1 phase. What all they have? So CDC6, CDT1 plus ORC forms a protein complex. This protein complex assembles with the MCM protein, which is a mini chromosome maintenance. So MCM acts as a helicase. See here you can see CDC6, ORC and MCM helicase is also present. Okay. MCM protein acts as a helicase. It is a hexamer of 6 related polypeptide, that is MCM 2 to 7, and forms a ring structure. It acts as a licensing factor which allows replication to initiate. After that, S-Cdk activation in late G1 phase. So S-Cdk is activated at the late G1 phase, triggering the assembly of several additional protein complex which lead to the formation of pre-initiation complex, which unwinds the helix and begins the DNA synthesis. As it initiates DNA replication, S-Cdk also, what does it do? It triggers the disassembly of some pre-replicative complex components at the origin. So it blocks the re-replication by triggering the destruction of CDC6 and the inactivation of ORC. It ensures that each origin is activated only once in each cell cycle. Here you can see assembly of new pre-replicative complex at origin, where APC bar C is activated, CDK is inactivated. So this was about the control of DNA duplication. So this table summarizes the major cell cycle regulatory protein. So protein kinases, where it adds phosphate groups, CDK activating kinase CAK, which phosphorylates the activating site in CDK. V1 kinase, which phosphorylates the inhibitory site in CDK, is primarily involved in suppressing CDK1 activity before mitosis. CDC25 phosphatase, it removes the inhibitory phosphate. So remember, V1 kinase and CDC25 are antagonistic in their roles. CDK inhibitor proteins. So SIC1 in budding yeast, P27 in mammals, P21, P16. Remember CIP, KIP family and INK4, I think. INK4. Yes, yeah, INK4 family, which are responsible for suppressing the CDK activity at different levels. Okay, and ubiquitin ligase, which ubiquitilates and responsible for degradation, where we saw this APC bar C, CDC20, CDH1, SCF. All these are helping in the regulation of activity of cyclin-CDK, where the major mechanisms we discussed, that is phosphorylation or dephosphorylation reaction, and also we discussed the APC bar C catalyzing the ubiquitination of regulatory proteins, and remember the activity of this APC bar C during securin and separase removal or dissociation, helping that separase to be active and help in breakdown of cohesin protein. So these are the major regulatory proteins which are regulating the cell cycle. So in our next class, we will be learning about the major topic, that is cell cycle checkpoints. So cell cycle checkpoints is the one which is very much needed to ensure that incomplete or damaged chromosomes and to identify them which are not replicated or which are already passed to the daughter cells. And these several checkpoints help us for to ensure the cell cycle to happen in a ordered way. So in our next class, we will be learning about cell cycle checkpoints. So that's for today. Kindly revise the topics. Thank you.